everybody. Today we are going to be talking about hearing as the second of the senses that we're talking about in this class. So hearing is also called audition, our sense of hearing, our auditory functions. And as we said, in order for information to be sensed, information from the environment, to be seen in terms of vision or heard in terms of hearing, we've got to take physical information from the environment and transduce it into or change it into action potentials which are sent to the brain. So in the case of hearing, we are transforming sound waves into action potentials so that we can hear. So sound waves are periodic compressions of air, and when we hear, it's a sound wave that's striking our ear. Sound waves vary in their amplitude, that means how tall they are, and their frequency, that means how short or long they are. So here we have amplitude being smaller sound waves versus higher amplitude are taller sound waves. And amplitude gives us the intensity or loudness of a sound. So the larger the amplitude, the louder the sound. And then frequency is the number of waves per second, and that's related to pitch. So here's frequency, low frequency or slow waves, or higher frequency or much quicker sound waves, and the higher the frequency, the higher the pitch that we hear. And we can hear in the range of 15 to 2,000 hertz. Hertz are cycles per second, so again, coming over here, one full cycle per second. That's what we're talking about. We're talking about a cycle. And our ability, we'll talk about, to hear the higher frequencies falls off with age. And people have a much harder time hearing these very high frequency sounds, or if you're exposed to repeated loud noises. So let's talk about the structure of the ear. There are three main sections of the ear that we'll talk about, and each section is made up of multiple parts. So the first is the outer ear. That's this part here, the part that you see when you're looking at someone's head. It's the cartilage that's attached to the side of your head. That's the outer ear, or we also call it the pinna. And it has this funny shape, but by having that shape, it's altering the reflections of sound waves and helping to funnel them into your ear. We'll talk about how this outer ear also helps us locate sounds. And then also considered part of the outer ear is this ear canal, or the auditory canal. This is an air-filled section of our ear. Then we go to the middle ear. I'm going to go back for 1 second, just so you see where we are. The middle ear is going to be this section here that I'm pointing to from the tympanic membrane. This is the eardrum, the bones in the ear, and then the oval window, which will be right here. So this is the part we're talking about in the next picture when we look at the middle ear. So here we are in that middle section. What are the structures that are in the middle ear? Well, the first is this tympanic membrane, or the eardrum. And the eardrum starts to vibrate, but the whole point of hearing is to transmit these sound waves into our inner ear and to turn them into action potentials. And so the tympanic membrane, this is the first place where these sound waves start coming in, and they hit this membrane, and the tympanic membrane starts vibrating at the same frequency as the incoming sound waves. So you can think about this a little bit like a drum pounding or beating at the same frequency as the sound waves that are coming into our ear. Then there are three tiny bones in the middle ear. They're right here, and they transmit this sound wave information to the oval window, which is right here. Okay, and so these bones start, the tympanic membrane is beating at the same frequency as the sound waves, which makes these bones, which are the first one here, connected to the tympanic membrane, starts to also vibrate at the same frequency as the sound waves, and all three bones start to vibrate or transmit that frequency information to the oval window. The oval window is much smaller than the eardrum, and what happens is when you are transmitting this information from a larger area to a smaller area, we basically are increasing the force of this beating or this frequency pulse on the oval window. And that's what we're trying to do. We're trying to get this sound wave information much stronger. And that's what happens here. And in this area of the middle ear, we also have two muscles that work to control volume. And so if something is very, very loud, these muscles are activated and they stiffen and they end up reducing the effect of the sound, okay, if something is too loud. So we've had sound waves that, I'm just going back for a second, go in through the outer ear, the pinna, through the ear canal. Remember, these are frequency or vibrating, kind of pulsing sound waves that then start to vibrate the tympanic membrane, or the eardrum, which then starts to vibrate the bones of the middle ear. And again, we start then with the bones of the middle ear attaching to the oval window and starting to have that oval window vibrating with increased frequency, with increased intensity at the frequency of the sound wave. And so we finally now get to the inner ear. Here's that last bone of the little middle ear bones on the oval window. And the inner ear is also called the cochlea. The inner ear is fluid filled and it's wrapped around like a snail. And when this bone starts to vibrate the oval window, the fluid inside the cochlea also starts to vibrate. And this cochlea has 3 fluid-filled tunnels, as you can see here. Okay. So now this is sort of a cross-section of the inner ear. The organ of Corti, and that's what you're looking at, this whole picture, is the part of the cochlea that converts finally sound waves into neural activity, into an action potential. So up until now, all we've had is the sound waves kind of vibrating all the parts of the ear, the outer ear, the middle ear, and the inner ear, but we haven't yet had an action potential. We haven't yet transduced that information into an action potential. And so what you'll see is we've got some main parts. We have the basilar membrane, this purple part, or we call it like the base basilar membrane. And we have hair cells. Here are hair cells. These are the nerve cells or the sensory receptors in the ear. So just like we had rods and cones in the eye, we have hair cells in our ear. These are the receptors that are going to change sound waves into action potentials. And we have up here, this is a tectorial membrane. This is a very stiff, hard membrane that's kind of right over the top of those hair cells. And remember, this is all in fluid, and the fluid is starting to pulse this organ, or start to pulse through at the frequency of the sound waves. And what happens is as the fluid moves across the basilar membrane, there's the base here, and the basilar membrane starts to move, starts to pulse at the same, again, some frequency as the sound waves, it starts to pulse. And so these hair cells move up during the pulses, and they hit this tectorial membrane, which is a hard membrane. And it's this hitting of the hair cells against the tectorial membrane that causes an action potential to happen. So here we have fluid moves the basilar membrane across the tectorial membrane. So the basilar membrane kind of moves, the tectorial membrane stays hard and stays in place, and we've got these hair cells that get pushed up against the tectorial membrane, and this excites the hair cells or the auditory receptors. We get a depolarization of the hair cells, neurotransmitter is released, and we have an activation. We have sound waves transduced into an action potential. That excites cells of the auditory nerve, and that information then goes to the brain, and we finally hear something. So going back to this picture, we have sound waves that are coming in through the outer ear or the pinna, through the ear canal. Those sound waves are making the tympanic membrane start to vibrate, which then makes the bones or the ossicles of the middle ear start to vibrate, which are attached to the oval window. The oval window starts to vibrate with increased intensity, which then causes the fluid in this cochlea or the inner ear to start to pulse as well. And again, we've got the basilar membrane starting to move as the fluid starts to move in the inner ear, which pushes these hair cells against the tectorial plate, which causes an action potential. And finally, we hear something. So as I said, sound vibrations cause the basilar membrane to kind of ripple. Imagine like shaking out a rug. And different parts of the basilar membrane. So here we have our cochlea that's all wound like a snail. Different parts of the basilar membrane respond to different frequencies. So the higher frequencies have the greatest effect here near what we call the base, where it's narrow and relatively stiff, and lower frequencies produce larger responses near the apex, where it's wider and more flexible. All right, once we've activated those hair cells and start to get an action potential, the auditory nerve fibers terminate in what's called the cochlear nuclei. So here we are in the cochlea, and it looks like we've got left cochlea over here in blue and the right cochlea over here in red. And then once the cells are activated, the message is sent to what's called the cochlear nucleus. Okay, right here. At this point, information crosses. You can see information from the left hemisphere is crossed, goes to the contralateral side of the brain for processing. Information from the right hemisphere, or from the right ear, is sent to the left hemisphere for processing, or we call that the contralateral hemisphere. So after information goes to the cochlear nucleus, then goes to what's called the superior olivary nuclei. It's already crossed by then. Information then goes to the inferior colliculus. That's part of the midbrain. We talked about the superior and inferior colliculus in the beginning of our class. This is the primary auditory center of the midbrain. You can kind of follow that information to where it is in the brain. After the inferior colliculus, information then goes to the medial nucleus, okay, of the thalamus. Remember we said the thalamus is the sensory relay center of the brain, so information then goes to the medial geniculate nucleus of the brain. And then to auditory cortex. Our auditory cortex is in the temporal lobe, just like we had visual cortex in the occipital lobe, and that is the pathway. So from the ear, from the cochlea to the cochlear nuclei, At this point, information changes or crosses to the opposite side, then up to the superior olivary nucleus, then to the inferior colliculus in the midbrain, then to the medial geniculate nucleus of the thalamus, and then finally to the temporal lobe in what we call auditory cortex. At every level, our auditory pathways have a tonotopic organization in that they're arranged in a map of low to high frequencies. And at higher levels, the auditory neurons are excited by certain frequencies and inhibited by neighboring ones. So that gives us the ability to discriminate and to hear tiny differences in sound. Differences in frequency of the sound waves are important for our sense of pitch. So frequency, that's the physical property of sound. Like we said, how quick or how slow the sound waves actually are. But pitch is our subjective perception of that sound. So how do we hear different sounds? Well, we've got two theories. We have what we call the frequency theory, and that is that the basilar membrane vibrates at the same frequency of the sound, causing the auditory neurons to produce action potentials at the same frequency. So at low frequencies, we have basically the whole basilar membrane in the cochlea vibrating at the same rate as the or the same frequency as the sound waves. And so in the frequency theory, basically you're saying that the entire basilar membrane starts to vibrate at the same frequency as the sound waves, and that causes us to hear sounds of that particular frequency. But neurons can't fire above 1,000 hertz or 1,000 cycles of a sound wave per second. So that's a problem of the frequency theory because we can hear at sounds above 1,000 hertz. And that gives us the place theory, which is that each area of the basilar membrane vibrates to a specific or a different frequency. And we already talked about this, the lower frequencies in the apex and the higher frequencies over here at the base. And so the current theory really combines both versions of the place theory and the frequency theory, that at low frequencies, sound is best explained by the frequency theory, that the whole basilar membrane vibrates to that same frequency. And high frequency sounds are best explained by the place theory. Audition in the cortex. Primary auditory cortex is where information goes first after it kind of leaves our ears and goes through those different processing centers. When it gets to the temporal lobe in the cortex, starts at primary auditory cortex, which is in the temporal lobe. And we already said each hemisphere receives most of the information from the opposite ear. The auditory cortex is specialized for detecting biologically relevant sounds like footsteps, animal sounds, speech. And this auditory cortex, its sensitivity is fine-tuned by experience during development. What we see in primary, and just like in vision where we had primary visual cortex, secondary visual cortex, etc., we've got a number of areas of auditory cortex. Primary auditory cortex cells in that area respond best to one tone, and cells preferring a particular tone are clustered together. Herschel's gyrus is part of the primary auditory cortex, and what we can see in MRI studies is that it is much larger in professional musicians than other people. So professional musicians who have spent a long time developing the sense of their sense of hearing and being able to discriminate amongst tones, you can actually see this difference in MRIs. Secondary auditory cortex, each cell responds to a complex combination of sounds. It's kind of another step further up in our processing of sounds. The book talks about amusia, which is an inability to discern tunes or to sing. associated with abnormalities in the right frontal lobe and poor connections between the frontal and temporal lobe. So that's an interesting disorder of audition. Hearing loss, about 99% of hearing impaired people have at least some response to very loud noises. There are different types of hearing loss. We have conductive or middle ear hearing loss, and this can be caused when the bones of the middle ear are unable to transmit sound waves, but these individuals have a normal cochlea and auditory nerve. So this can be caused by tumors or infections or disease in that middle ear area, and so it stops that transmission of the sound waves from the outer ear to the inner ear. This can usually either be corrected by surgery or with hearing aids. And interestingly, people with middle ear deafness can hear their own voice because your own voice actually bypasses the middle ear and just vibrates the inner ear directly. If someone has nerve deafness or inner ear deafness, also called sensory neural deafness, this is when there's damage to the cochlea or the hair cells in the cochlea or the auditory nerve. This can be treated with hearing aids if there's not extensive damage. It can be caused by genetics, disease, repeated loud exposure to loud noises, etc. And sometimes when people have this sensory neural or inner ear nerve damage, they have tinnitus, which is a ringing in the ears. This is very common in old age because individuals in old age often lose the high frequency hearing, or you can get this after damage to the cochlea. And what happens is other axons, other neurons may actually invade the area in the brain that's responsive to sound. So if that part of the brain isn't getting any more information from the cochlea, from sound, then areas around it may start to innervate or may start to take over that particular part of the brain, and so you end up with kind of a ringing sound in your ear that is actually not, doesn't have anything to do with actual sound waves being produced. And this is similar to mechanisms of phantom limb, which we'll talk about when we talk about the motor system. And then we also have central deafness. This is damage to auditory brain areas, like a stroke or a tumor or traumatic injury to the brain. So the ears are working fine, inner ear, middle ear, etc., no problem there. But when you have damage to certain brain areas, again, that can impair hearing. And can impair different aspects of hearing. In fact, some people can have what we call word deafness, which is particular difficulty recognizing normal speech sounds. They have normal speech, they're still able to speak. We think speech production is located in the frontal lobe, so still have normal speech production, and they can still hear nonverbal sounds, but difficulty understanding words, basically. And cortical deafness, this is difficulty recognizing all complex sounds, verbal or nonverbal. This is very rare. What about localization of sound? How do you know where a sound is coming from? And if you were to close your eyes and just listen to the sounds around you, not only would you hear the sound, but you would know where the sound is coming from. How do we do that? At higher frequencies, we believe your head creates what's called a sound shadow, or your brain is basically looking at intensity differences between the sound coming at your right or your left ear. Basically, the sound is loudest in the ear that is nearest to the sound. So here we have a sound wave that's hitting the left ear, which is closest to the sound first, your head creating this sound shadow, and the sound waves going to the right ear are less intense. And so the brain does this calculation of comparing the intensity between the ears and can say, oh, the sound must be coming from this left side. In order for this to happen, the wavelengths must be shorter than the width of your head. But at very low frequency sounds, we can determine localization by what we call phase difference or latency difference, that sound arrives out of phase depending on where the wavelength is compared to the width of the head. So looking here, if we've got sound waves that are coming from a particular direction, and you're hearing each ear is getting a different at a different phase in the wave, in the sound wave, will hit the ears at different times. And so again, the brain does this calculation of the difference between where in the phase of sound it hits one ear compared to the other. And by doing that, our brain is able to tell us where the sound is coming from. Okay. Last thing we'll talk about is the vestibular system. It's not part of your hearing or sense of audition, but it is in that area inside of our, right near our ears. So the vestibular sense responds to the system that detects position and movement of the head. It directs compensatory movements of the eyes, helps us maintain balance. And basically, we have these three semicircular canals that are, you can see inside. I want to see. There they are. Right in here, three semicircular canals, and they are filled with a jelly-like substance. And basically, they also cause bending of hair cells that cause action potentials. So taking basically movement of your head, and depending on the movement of your head, causing an action potential that that our body and brain uses to help us maintain our balance. And so the canals are oriented in the three different planes of your head movement: nodding, so kind of that y-axis, shaking your head, and tilting of your head as well. So all three different ways you can move your head. We have these three different semicircular canals that, when the jelly-like substance inside moves based on the way you're moving your head, it causes action potentials and helps us compensate with our eyes and our body movements. So head movements initiate flow of this fluid through the semicircular canals on the same plane, signaling movement to the brain. And we have specialized receptors actually as well that provide information about acceleration and deceleration. So are you moving forward? Are you slowing down? All of this information important for keeping our sense of balance. And many of these nerve fibers terminate in what we call the vestibular nuclei in the brainstem, and some go directly to the cerebellum. Cerebellum is also really important in balance. Perhaps motion sickness can result from too much vestibular excitation or a conflict when you're receiving contradictory information between your visual sense as well as your head movements. And so that conflict can cause motion sickness. All right, thank you very much, everybody. Bye-bye.